So now I'm going to switch gears a little bit and move on to the project grant applications themselves. Congratulations to all of you who have been invited. As you'll see in a minute, you've got past the first big hurdle as we've narrowed down to a much smaller number of project applications. Here we go. So we were very fortunate to have 107 pre-proposals. These were reviewed by our leadership team that you just saw on the previous page, as well as our internal advisory committee that has representatives from all the universities involved and the state. And we have whittled those down to 16 full proposals. And so this is where you are right now. We've got 16 full proposals, and those will go forward. As you know by now, there's a fairly detailed 12-page application form, as well as an expected for you to give a pitch to our external review board, which is filled with representatives from pharma and medical device companies, from investors, and so on. And we chose that number specifically to balance, you know, wanting to have the most technologies possible and being able to help those innovators, but also not to have too many that we have too many that we can't manage to help. And so this is a very hands-on process. We feel that this is very much experiential learning. By completing the full proposal forms, we hope that everybody involved will learn a lot about the product development process, and we'll get a lot of great feedback on how best to develop their technology that they will use going forward, so that once you've learned how to do it one time, you'll be able to do it over and over again. And a significant, you probably got about a 50-50 chance of getting through to the next round, and a good chance of getting funded. But we hope that everybody, regardless of whether or not they end up getting a funded project, will find this of great value. Okay, so what do you need to know about these KYNETIC grants? The number one thing, and the number two thing, and the number three thing is that they are not like traditional grants. A lot of us are used to writing grants such as NIH R01s, and these are very, very different grants. When we see your application, we don't want to hear about hypotheses or things that you're doing to discover something. We want to hear about the product itself. So we want you to start kind of at the end and work backwards. What is the ultimate goal of the research that you plan to do? This is not like an NIH grant, usual NIH grant, in that we don't just give you the money and leave you alone and ask you kind of to tell us how it worked out at the end. This is a managed process where we will be checking in on you. We will be, our project managers will be an inherent part of your team to help guide you through the process of product development, make sure that you stay on track and use the money for what it's intended for. As I said before, you can get up to $200,000 in total funding for a project. However, you have to compete each time. It's not guaranteed, and it will be dependent on not only completing the milestones a lot of times that you've set for yourself, but also in competing with other technologies that are out there. And this is not just, we don't do this just because we're mean people, although I might be, I don't know. But NIH will be watching us to make sure that we do this. This is really the key difference that they really want us to take, because it makes things move much, much faster. The other thing that's different is that we are allowed to spend money on things that are not usually allowed on NIH grants, such as consultants or contract research organizations. So we don't mandate those things, but those things are allowed. Okay, so some of you have probably looked at the full application forms and started screaming. And I don't blame you. I've looked at those applications form. I've helped many teams fill them out. And I agree with a lot of complaints that we have heard. So the reason that we have these forms is that the NIH wanted a standard format that contained all the necessary information to allow their panel of experts to know everything they need to know about your technology. So I kind of glossed over it in the first slide of this kind of tips part, that after the external review board comes the NIH Technology Guidance Council, and that is populated by experts from agencies such as the Patent Office, the FDA, the CMS, which is what decides Medicare, Medicaid reimbursement. And so these are the real experts. You're getting just a great amount of knowledge really from the horse's mouth in terms of feedback on your proposal. And so we even though you know, the forms are a little bit repetitive and a little bit confusing, this information is really necessary to get the best kind of feedback that will be useful for you. And the other thing I've heard, you know, a lot of people, and even some of the other universities that are doing this, are saying, well, you know, let the scientists do the science. The technology transfer officials can help with the IP part. We can get our staff to fill out all the other bits. But we try to kind of make a balance between helping you, but at the same time, not kind of just giving you the information. We want you to learn how to find the information. And so we'll be there to help you. We don't want anyone to be scared or worried about this. We'll help you through the process, but at the same time, we're not going to fill out the forms for you. We want you to understand, you know, what is involved, and really, you know, have to do some work to understand this. So that kind of fills out the bottom part too. And the reason that we do this is, you may not think it's necessarily to know all these things. You know, we've heard, why do I need to know all this IP stuff, regulatory stuff, reimbursement stuff? I'm not going to be doing this. I just want to get this so I can license it and a company can deal with all that stuff. And that's true. And we don't want you to necessarily become experts in the business side of things and the IP side of things. You don't have to become experts in any of these things, but it's important that you understand enough about them to be able to kind of map the path of from your idea or your discovery to an actual product and to the market. Okay, so I'm going to give you, I've given you, all of you should have received kind of a list of tips that were written by me. I'm not going to go through each of those. You can read those. But I wanted to just give you my perspective. So I am like probably a lot of you. I'm not a business person. I call myself a reluctant entrepreneur. I don't have an inherent interest in business, but I've founded a company, I've worked with industry, I've applied for commercialization grants, and I've done this because my primary interest is seeing what I've discovered or an idea that I have get to patients. And, you know, the truth is that unless you understand a little bit about the business side of things and know about commercialization, it's very, very difficult for you to move your new innovation and turn it into a product. And so my kind of, I don't have any formal qualifications in business. I'm kind of pretty much making it up as I go along, but I think it's hopefully an educated you know, opinion, in that I have directed the Excite Reach Hub at U of L for the past 5 years. Between us all, we've reviewed over 170 applications and have managed 26 projects, and many of those have gone on to be successful. Most of our projects have either been licensed to third-party companies. A lot of teams have formed their own companies to develop the technologies, and a few of them have been terminated. They figured out that their idea was not going to work, and we consider that a success too. And so now, as you know, I'm co-director of the KYNETIC Hub, and I've also been a reviewer for both of these programs, but it's important to understand that I'm one of about 20 reviewers that will be assessing your proposal. And so, you know, these are my own opinions. Okay. So just general things to think about when you're writing your proposal. Again, I don't want to hear about hypotheses or specific aims. I want to hear about your product. Very specifically describe to me what your product is, who will use it, how will it be used, and so on. We want to fund projects, I think, that where the funding will have an impact. So try to explain to the reviewers why having KYNETIC funding is important for you to get to this next level, rather than being kind of an incremental advance. be realistic. Don't tell us that, you know, in 2 years you're going to have a drug to market and it's going to make $1 billion, because we won't believe you. So really try to be realistic about what it takes. And I don't think I see the project managers are smiling, so I don't think they're going to let you say things like that. But the other thing is, you know, be as specific as possible. I don't want to hear that, you know, this is a great project, this is a great product, it's going to be better than what else is out there. I want you to show me, you know, a table of all the other competing products that either exist or are in development, and little, you know, check boxes for their specifications versus your specifications, and show me very explicitly why this is going to be better. And then the other kind of tip is, remember who your audience is. So again, this is not an NIH grant going to a study section of experts in your very narrow field. This is a product development grant that will go to a panel of external review board members who are a mixture of scientists and business people. So I always tell people, try and explain what you want to do and why you want to do it, as if you're telling your, you know, highly intelligent but non-scientist neighbor. And be concise. At some point in this process, you're going to be asked to give a 5-minute elevator pitch describing what your product is and why you want to develop it. And remember, this is, you know, it's a mixture of business and science. It's very easy to describe why an Envision product is very useful for patients, but also you need to describe why it's a good business proposition too. And then finally, you know, we're here to help you, but we're not going to give you the best feedback if you give it to us, you know, the night before it's due and ask us to look through this 12-page proposal. So make sure you kind of work with us and give us plenty of time so that we can do an iterative back and forward on what your application will look like. And so I recommend that before you even get started writing, you meet with project managers or talk with project managers about you know, where you're headed, but do as much as you can by yourself first, and then we'll get together and talk about it. Okay, so these are the things I've already said mostly. I want to hear about the product, what the need is, how many people will use it, how's it better than the way things are done right now. And here's something that's a little bit different too. I want you to think about stakeholders, okay? I want you to think who is going to be affected by my product. So in biomedical product development, you know, the patient is the end user. Sometimes the patient's the customer. You might be selling directly to the customer, but a lot of times the insurance company is the one who's going to pay, or the hospital is the one who's going to buy your device. And then the doctors or the nurses or the dentists or the clinical providers are the ones that are going to use it. So you have a lot of people that you need to persuade that your product is a good idea. And I want you to think about who these stakeholders are and why they're going to want to use your product rather than what's out there or whatever the alternative is. Okay, and then the other thing is that this has to be feasible. That you have to be able to make it, you have to be able to scale it up and manufacture it on whatever scale you need. And you have to be able to break into the market, and you have to have a way, you know, for example, if it's a treatment, you have to have a feasible way to run a clinical trial and get the answers that you need. So, you know, for example, if you were developing something to treat, you know, a disease that takes 20 years to show up, that's not going to be practical to do a clinical trial in that. So I need you to think about how do you get to FDA approval. Okay, and so if you haven't figured it out by now, you know, this mechanism requires you to think very differently about research. We want you to start at the end and work backwards. You know, you see a product, for example, a new treatment for non-small cell lung cancer, stage 3, non-resectable. I want you to work backwards and say, you know, what is the clinical trial I need to do to get this approved? What FDA regulations do I need to go to? How am I going to get this to market? And almost certainly in the case of drugs, it will at some point require you to partner with a pharmaceutical company or license it to a pharmaceutical company. And the other thing, the final thing, very important thing, is intellectual property. you may have found, you know, a plant in your garden that might be a great treatment for something, but natural products are not generally patentable. And so if there's no way to patent something or somehow protect it from copycats, that might be a difficulty in terms of development. And then the other thing that's very different is, in a lot of cases when we write NIH grants, even though we say we're testing hypotheses, really what we mean is we're trying to prove ourselves right. We're trying to prove that our idea is correct. And so this is differently. We want to prove that we're wrong as quickly. We want to find out if we're wrong as quickly as possible. So rather than kind of building up to the big experiment, we want to go straight to that big experiment. We call it the killer experiment. It's the experiment that you can do that's going to kill your project. It's going to prove that your idea is not going to work as quickly as possible. And really that's a success. And so that's what we're looking for. We're looking for you to do that big experiment that will prove whether this will or won't work, or it won't prove it will, but might prove it won't. Okay, and while we understand that, you know, conferences, publication, and grants are always important for academic scientists, we all need to do these things, we don't really want to hear about how this is going to lead to a new paper in science or a new grant application. That's not our focus. Our focus is the product. But, you know, if you can convince us that presenting at a conference or publishing in science will get you a lot of attention and make it easier to license your product, we might consider it. Okay, and so these are, again, all the things that I just said. I'll let you read through those in your own time, but I wanted to point to some of the reasons why, even though you might have great science, you might have a great idea, we might have a lack of enthusiasm for it. So a lot of times it might be that a similar product already exists. First thing you should do is Google your envisioned product. Make sure somebody hasn't already invented it and developed it. Again, patents, intellectual property, it doesn't have to be a patent, it could be a copyright, but there has to be some way to prevent people from just, you know, you spend all this money and time developing something. If someone can just come along and copy it and make the same thing, you're not going to have a very good business model. Again, we might not be excited if it's just a little bit better than what's out there. We're looking, not always, but a lot of times we're looking for things that will have a really kind of major impact, both in terms of, you know, on patients and on the market. We might not have enthusiasm because it seems, even though it's a good idea, we think it might be unlikely to work or impossible to tell if it works. And then there are other times where, you know, we think this is a great idea. This would really help patients a lot, but you know what? It's never going to make any money. So how are you going to get someone to pay the hundreds of millions of dollars it's going to take to develop it? So, you know, we don't have to make money, but it's very hard to develop something or to get investments for things that are not going to make money. So if you're planning on something that is not going to make potentially a lot of money, then you have to kind of have an alternative plan, such as a foundation or a philanthropic organization. Okay, so how do you find out all of these things? This is the tough part. And you know, again, this is just things that we've picked up as we go along. If you have other ideas, please let us know. We'll add them to our list of ideas. So the first thing to do is just Google it, right? Just you know, look up all of these things about your product, and it maybe will give you some information. Look on clinicaltrials.gov, see if there's anything like that that's already been tested. And then other resources, you see the list here, I'll let you look through it. You know, the usual places that we're used to looking, such as PubMed and Google Scholar, but also patent databases. A lot of times FDA has issued notices about certain products and how to get them approved or what can and can't be done. But then the things that are highlighted here in orange are the things that I've mentioned already, these slides themselves and the tip sheets that go with them. But really our staff are some of your best resources. They're going to sit with you and spend time to meet with you and go through those application forms. But also, you know, we're researchers, and we a lot of times are located in hospitals with and in universities with business schools. Let's take advantage of all the people we can. I mean, get outside of your comfort zone, leave your lab, go find somebody in the business school to ask questions, go find somebody in the hospital. Maybe they might be a bit busy right now, but at the end of the day, when things come down, find clinicians that have time to talk to you about your product. Okay, and then just to wrap up, I wanted to tell you, what are we trying to achieve here with this KYNETIC? This is the kind of big picture of what we want to do with this program. So we want to impact as many people as possible in a positive way. We are deliberately trying to cast a wide net to find all those exciting technologies out there to really, pick your brains and find out those good ideas you may have. And so that we can find, fund, and nurture those projects with the highest potential. We want to have a lot of different types of innovations in our portfolio. We know that some things are high risk, high impact, and other things are, lower risk and maybe not so much of a big impact, but could get to the market fairly quickly. And so we want to try and kind of balance those, that just makes good business sense. We also want to recognize that, there are a lot of different types of innovators out there. In our previous REACH program, we had everyone from, senior professors to graduate students, postdoctoral fellows, staff. We want to reach all of these people, and we understand that a lot of times, professors are not going to give up their day jobs and go start a new business based on their technologies, but maybe graduate students are, maybe postdocs are, maybe staff are. And so we really want to encourage you, and even if you're not willing to do that, just learn this early on in your career so that you can have it as you move forward. And even if this current idea doesn't work out, next time you come across a great idea, you'll know what to do with it. We want that everybody who participates in this program will benefit from it. One of our key features is that we try to give you feedback, honest, constructive feedback at every step of the process. So those of you who put in pre-applications, you should be getting very soon, if you haven't already, some feedback on that. And similar with the full applications, and with the, if you get to the NIH Technology Guidance Council, you'll get feedback from the FDA and the Patent Office and all that alphabet soup of federal agencies. And we want to stay engaged with you throughout the program. Just because you didn't get funded, or just because your project's finished, we still want to hear from you. We still want to interact with you. We still want to hear you come to us with ideas for future applications. And so how do we know if we've been successful? Well, there are obvious tangible things that we can measure: the number of patents we get, the number of licenses we achieve, the number of companies formed, the follow-on funding. These are all tangible things we can measure, and we will. But also what we've seen with our previous experiences, that there are a lot of kind of intangible impacts, which are a little bit harder to measure, but really are apparent very quickly, and really can be some of the most impactful things. And these are things like changes in the mindset of researchers. And you see my little cartoon here where, of course, we still want you thinking about grants and publications, but we want you to be able to think in this different way about this different type of research that is very complementary to maybe your existing programs, but where you're focused on turning an idea that you have into a product, and that the things that are involved in that, and that you want to move very quickly towards developing that product in parallel with your traditional research. And also we want to have this same kind of impact on the thinking and the institutional culture. So we want universities to start valuing this type of translational research in both, congratulating researchers for this, and in terms of promotion and tenure, considering things like patents and maybe SBIR grants and things like that. So, and finally, I wanted to say again, emphasize that this is a statewide program. One of the most exciting opportunities I see is the potential for collaboration between institutions, between UK and U of L, between these bigger institutions and the regional universities and the community and technical colleges. I'll be really excited if we can have interactions with all of those regional universities and technical colleges, and get people involved in both the project funding and the training, and just being engaged with getting excited about doing translational and commercialization focused research. So with that, I'll finish up, and thank you for listening. I'm gonna be available to answer any questions from our team now if they have any, and you can always email us, and your project managers will let you know the email for that. My email is paula.bates@louisville.edu if you want it straight from the source. We have emails that are KYNETIC@louisville.edu and KYNETIC@ukentucky.edu. But your project managers will be in touch with that information. Thank you.
Paula, one of the questions that applicants have asked us already is, how should I approach coming up with what my killer experiment will be? Can you talk a little bit about how you as a researcher would think about that question, and approach that to decide what their initial killer experiment would be?
Sure. So, and it's important to emphasize there's not just one killer experiment. I think the way you want to think about it is, what are the risks of this product, right? So I'll give you an example of a small molecule drug development. So, if I've found this great target and I have a prototype inhibitor, and I want to do some medicinal chemistry to find a lead compound and then develop it into a potential drug, firstly there's the risk that I might not be able to find a more active compound, and so my first milestone will be to find an active compound that is has an IC50 value of say 100 nanomolar or whatever, or be specific in your milestone. So that's a milestone, and then when we think about why drugs fail to be successful, firstly there's toxicity. You might have a great compound that inhibits an inhibitor, but it's extremely toxic. So, a killer experiment, and maybe this is a bad choice, but would be to test the toxicity in an animal model. And if you see severe toxicity at similar concentrations where you're going to, need to be treating for activity, then that's it. This inhibitor that you developed is no good. You're going to have to go back to the drawing board and try something different. The other major reason why drugs don't get through the clinical trials process, don't get approved, is because they don't work very well. And so again, a killer experiment would be to see if this treatment that you're proposing works in an animal model of the disease. And again, up front, before you go into it, specify what activity is acceptable. If I see a 50% reduction in tumor growth at 10 milligrams per kilogram, that's satisfactory. So don't just do the experiments and see how they come out. Decide upfront what is an acceptable kind of cutoff, and that's what we mean by having those milestones. And the go no go decisions are, if it's more than this, then I move on to the next experiment. If it's not, either I stop the project or I go back. And it's important to think as well, milestones are not just experimental, they can be things like patents. And so a lot of times somebody could have been doing great, the science is going great, but maybe they don't get a patent application allowed that they were expecting or needed, or they found out there's another patent that is very similar. So a lot of times we may end up, having to say, well, a project's not viable, just not anything that the researchers did, but just circumstances.
That's great. Thank you.
And just another example, I do drug development, so I talk a lot about how it works for drugs, but, again, with devices, firstly, you've got to think about, can I manufacture this? Is it, the risk factors is you can't scale it up, or you can't manufacture it. There could be a certain throughput that you need, or there could be certain, specifications that you need it to perform to. So those would all be kind of killer experiments. Can I make this? Does it perform to my, predefined specifications?
Excellent. Thank you. Anybody else have follow-up questions?